Hello, Senior 2. Now we are going to explain lesson 2 about photosynthesis process in green plants. Okay, photosynthesis process is very important process because it share and help a plant to make its food. Making food simply by conversion of simple components to complex components. Simple components, inorganic components like water and the carbon dioxide, and turn them to organic components like carbohydrates, glucose, from low energy components to high energetic components. So photosynthesis process is very important process for plant, and every living organism make its process. This process. Also oxygen is gas which result by side of carbohydrates. So we have two products here. Number one, and the essential one, glucose, carbohydrates, and another one is oxygen. Okay, the raw materials. One, water. Water is the first inorganic components we need to make photosynthesis process. Water, okay, is the production of carbohydrates. Mineral salts, as we said before in lesson 1, mineral salts, we have two types of minerals, macro and micro. From macro, okay, nitrate, phosphate and sulfate. All of them can change into lipids and protein from carbohydrates, which result from photosynthesis process. Phosphorus, okay, is very important because without it we cannot obtain energy, because phosphorus, the main or share in the compound of ATP that used in storage of energy. Also magnesium. Magnesium is very important element because it share in the structure of chlorophyll. Chlorophyll consists of one atom of magnesium, but this atom, okay, hypothesized by the scientists that is responsible for absorption of light. Iron, the last one, iron share in making enzymes, and any biological process like photosynthesis process and any process occurred inside living organism cannot complete its mission without the help of enzymes. Iron share in making enzymes. And the second here carbon dioxide. Carbon dioxide, okay, only source from which green plants obtain carbon. Okay, products of photosynthesis process, as we said, number one, glucose, carbohydrates, so it is simple sugar from monosaccharide. This monosaccharide later may be burned or share in cellular respiration and give energy, or share in building to give protein and lipids for growth. So monosaccharide, okay, has two paths. Number 3, or the last path, is helping in storage. If the amount of monosaccharides that were resulted from photosynthesis process a huge amount, I can store them in shape of starch, okay, inside chloroplast. The byproduct or the beside product is oxygen. Rate of photosynthesis process. Meaning of rate, it is the speed of reaction. We can measure the rate or the speed of reaction by two products. First one, amount of oxygen. If amount of oxygen high, so the amount or the rate of photosynthesis process is also high. If number 2, the amount of carbohydrates, okay, was high, also the rate of photosynthesis process was high. Site of occurrence of photosynthesis process. Photosynthesis process occurred inside organelle. This organelle is named by chloroplast or green plastid. Green plastids or chloroplast present in one of two, maybe in green plant and also in green herpetious stems. Green herpetious stems contain type of tissue named by chlorhydrous tissue that contain chloroplasts. Structure of the plastid. By the diagram, easily can explain its structure. Chloroplast, by the light microscope, as you see here, has shape of convex lens. But under electronic microscope we can know the internal details of the chloroplast. Chloroplast consists of two side membranes, outer one and inner one, outer membrane and inner membrane. And look carefully inside the chloroplast. We have here discs. The group of discs is named by granum. Granum. One of those is thylakoid. It is not your process, but it's named thylakoid. Between thylakoid and another thylakoid there is a membrane that may extend between them. It's named by lamella. Inside discs or thylakoid contains chlorophyll. Outside of granum, there present not white, not empty, but it's colorless. Why colorless? Because containing of proteins, and the protein is colorless. It's named stroma. So we have here two parts. The inner one, discs one, granum, and the outer one is stroma. Inside the granum we have pigments that help in one appearance of the color. Number two, help in photosynthesis process by absorption of light. Types of the pigments. First one, chlorophyll. Chlorophyll classified into two types, A and B. Chlorophyll green may be pale green or dark green. A is dark green or blue green. B is pale green or yellow green. Both of them share about 70% of the total pigments that present inside a plant. Dansophile and the carotene, both of them yellow, but dansophile lemon yellow, about 25%. Carotene is the color orange yellow, about 5%. Each granum consists of 15 discs, may more, at least 15 discs. And by increasing the number of discs we can or the plant can absorb amount more of energy. So plant dominant in green because about 70% of the total pigments chlorophyll, and the chlorophyll's color is green. The role of chlorophyll. Structure of chlorophyll: 655 H72O5, nitrogen 4, and only one atom of magnesium. Chlorophyll, by this atom, magnesium, can absorb light and turn them into ATP. By the help of ATP, he can complete his mission and form carbohydrates. So scientists hypothesize that magnesium atom is responsible for absorption of light. After making carbohydrates, we have problem here. The first Glucose. Glucose is monosaccharide soluble in water. Okay, I cannot depend on it to store it for later. I will change it into starch. So the rule of starch for storage carbohydrates till be used later. The structure of leaf consists of three main positions. First one, upper and lower epidermis. Number two, mesophyll tissue. Number three, vascular tissue. Here, TS in dicot leaf, meaning of TS, transverse section in dicotyledon plant leaf. Okay, by focus here we can see upper and the lower surface. Okay, upper and the lower surface is epidermis. Okay, epidermis is covered with cutin or cutin. Why? Because cutin is wax that cannot permit water to pass through, so it will reduce the amount of water that will be lost with the transpiration. Okay, leaving area that's named by stoma or stomata. Second layer, okay, it is mesophyll tissue. Mesophyll tissue, okay, is classified into two parts, upper part and the lower part. Look carefully for the upper part. Its name is palisade mesophyll tissue. Palisade is columnar elongated cells. Okay, and adjacent to each other, no intercellular spaces. Chloroplast here more than the lower one. Why? Because the upper palisade face to light. So we need here a big amount or a large amount of chloroplast to absorb light. Chloroplast here more. Okay, and concentrated where? At the upper part of the cell. Look here to the second tissue in mesophyll is spongy mesophyll or spongy layer. Spongy layer, irregular in shape, contain also chloroplast, but in fewer amount than palisade. Why? Because it distant from light, need less light. Okay, but the upper one need more light because it face light. Okay, there is intercellular spaces for aeration. This is upper, lower and the mesophyll tissue. The last one, this circle is the rib of the leaf, mid-rib of the leaf. Okay, we have here cycles. These cycles, okay, is named by xylem. Xylem is vascular tissue that's responsible for what? Transportation of water and mineral salts. Xylem face upper epidermis, while the lower one, the lower tissue is phloem. Phloem face lower epidermis. Its function, translocation of food. What's food? Carbohydrates after being formed by photosynthesis process and translocated to all plant parts like stem, like root, flower, grains, any part of the plant. This is named by vascular bundle. Okay, we explain all upper mesophyll consists of palisade and spongy layer. Palisade layer, okay, موجودة فين؟ below upper epidermis. Palisade possess many chloroplasts arranged at the upper part of this cell. Spongy layer, irregular shaped, have large intercellular spaces. Spongy, less in chloroplast than palisade layer. Vascular tissue, midrib contain main vascular bundles, while veins and venules contain many smaller secondary vascular bundles. Inside vascular bundle, as we said, we have two, xylem and the phloem. Okay, xylem toward upper part of the surface, okay, while phloem toward the lower part. Mechanism of photosynthesis process. Okay, please listen carefully to the next video that explain the mechanism of photosynthesis process practically. Thank you. Mechanism of the photosynthesis process. Okay, first, in determination the mechanism of photosynthesis process, okay, I have to know the source of oxygen that released from photosynthesis process. Okay, Van Nel, scientist, the first scientist, pointed out the role of the light in photosynthesis process, and he was his experiments on two living organisms. First one, sulfur bacteria or green purple bacteria. It's autotrophic bacteria, make photosynthesis process by bacteriochlorophyll. Okay, named by sulfur bacteria because these bacteria live in swamps and bones that reach in hydrogen sulfide. Okay, hydrogen sulfide here like water. Okay, source of hydrogen to reduce carbon dioxide, and sulfur will precipitate. Van Nel, please focus back here. Okay, light decomposes hydrogen sulfide into hydrogen and sulfur. Okay, first equation. Number two, this hydrogen used to reduce carbon dioxide by naming of dark reactions. So light or photosynthesis process is divided into two parts. First part, light reaction. Second part, dark reactions. Light reactions, decomposing of hydrogen sulfide, okay, into hydrogen and sulfur, while hydrogen latter used in dark reaction to reduce carbon dioxide to form carbohydrates, the simplest one, glucose, then be complicated into starch. So the equation of photosynthesis process: carbon dioxide, hydrogen sulfide, by the presence of sunlight and the bacteriochlorophyll, okay, turns to first product, the main product, C6H12O6 glucose, and water, then sulfur precipitate. On this basis, Van Nel assumed first one, light reaction, as we said, okay, similar to the plant, okay, but different from H2S, hydrogen sulfide, by water in green plant. Okay, so water here decompose in green plant in light reactions to hydrogen and oxygen gas. Oxygen evolve, okay, while hydrogen here is used in reduction of carbon dioxide in dark reactions. Okay, this reaction don't require light, so it's named by dark reaction. Okay, so Van Nel proposed that the release of oxygen comes from water, a case which is similar to sulfur that released from hydrogen sulfide. So the general chemical equations that represent the photosynthesis process in green plant like this: carbon dioxide react with water in the presence of light, and the chlorophyll here, not bacterial chlorophyll, produce glucose, water, and oxygen. Okay, to prove and the verification of the theory of Van Nel, okay, we had a group of scientists from California University use algae named by chlorella algae. Okay, this experiment prove what? Prove that algae condition, okay, favorable for photosynthesis process, and water is the source of hydrogen, okay, and source of oxygen, okay, not carbon dioxide. To prove this experiment, okay, we will use isotope of oxygen. Oxygen may be normal, so its atomic mass is O18, and the isotope 1O18. Okay, they discovered that oxygen which evolved during photosynthesis process of the algae was isotope oxygen 18. Oxygen 18 similar to oxygen of the water, okay, while the oxygen that's present inside carbohydrates and the water that product from photosynthesis process are same of oxygen of carbon dioxide. Okay, first experiment here, okay, carbon dioxide here, oxygen 16 ordinary, while water 18 isotope. Look here, glucose, oxygen 16, so ordinary like carbon dioxide, and oxygen in water also 16 ordinary like carbon dioxide, while oxygen that evolved from photosynthesis process like water 18 isotope. Second experiment, okay, by replacing oxygen and hydrogen by each other, okay, oxygen in carbon dioxide isotope while water ordinary, okay. Look here, oxygen that evolved from photosynthesis process also change like oxygen of water. That's mean oxygen evolve from water by break it down, okay. Light reaction and dark reaction. The scientist who explained light and the dark reactions of photosynthesis process was Blackman, okay. First one light reaction, second one dark reaction. Light reaction, as we said, okay, light the limiting factor, okay. Light reaction take place in grana. So we have here two informations about the light reaction, that light here is the limiting factor that determine the reaction, while light reaction take place inside grana. Number two, dark reactions, okay. Dark reactions are sensitive for temperature and they're not affected by light. This reaction takes place in both light and darkness. Why? Because here temperature is the limiting factor, not light. Okay, why temperature here is the limiting factor? Because the work inside stroma according to enzymes, okay, not light. Okay, light reactions of photosynthesis process. First, okay, when light falls on chlorophyll or grana, inside the chloroplast, some electrons in the atom of chlorophyll, okay, hypothesized by scientists this molecule in magnesium atom, okay, this electron will gain light and be excited and move to upper energy levels. To back to the normal energy levels, okay, have to lose this energy. So the kinetic energy is stored as potential chemical energy in the chlorophyll. Molecules of chlorophyll are therefore said to be excited or activated. When this stored energy, okay, is released from the electron will back again to the energy level, the normal energy level, okay, and this energy will be used later in the second steps in the photosynthesis process. Okay, so the energy here will be divided into two parts. First part, okay, energy stored, okay, in a molecule of ATP. Okay, second one, the second part, okay, will used in splitting water up into hydrogen and oxygen. Okay, oxygen will evolve and hydrogen will turn to dark reaction. So two parts here. First part, split water into hydrogen and oxygen. Another part, okay, will be stored in molecule of ATP by formation or by combination between ADP and phosphate B. This process is named by photosynthetic phosphorylation. Okay, so ADP plus B plus energy, okay, will produce ATP. The hydrogen here in grana, okay, to get out from grana to cythroma, okay, have to combine with a coenzyme. This coenzyme is named by NADB, NADB. NADB, nicotine amide, okay, dinucleotide phosphate. Okay, this coenzyme, okay, will react with hydrogen, or will deliver, not react but deliver hydrogen to dark reactions, so it will turn from NADB to NADBH2. Okay, the function of NADBH2, keeping hydrogen from evolving like oxygen. Okay, so it will keep hydrogen from escaping. Oxygen release as byproduct or secondary product from decomposition of water. This diagram show the mechanism of the light reaction. Sun, source of light, okay, give light energy, absorbed by electrons in chlorophyll A, and converted this electron or atom to activated chlorophyll, and after a while this energy will be released and it will be divided into two parts. First part is split water into hydrogen and oxygen. Hydrogen will be carried by coenzyme NADB into NADBH2 that carry hydrogen to dark reaction, while another part of the energy, it will turn ADB, okay, ADB to ATP by combination with the phosphate group. Okay, this process is named by photosynthetic phosphorylation, and carry also this energy to dark reactions. So we have here two products from light reactions will turn to dark reaction. First NADBH2, number 2 ATP. Okay, this also diagram show us the relation between grana and cythroma. Okay, the result here from splitting of water, half of oxygen and 2 atoms of hydrogen. Okay, this is a product here. Okay, PC1, PS2, those types and degree of chlorophyll. Okay, it is not your business, but here from this diagram I want you to know, first one, the splitting of water, number 2, phosphorylation, photosynthetic phosphorylation, okay, by reaction between ADP and B and formation of ATP. Okay, those will go to cythroma. Okay, dark reaction of photosynthesis process take place, as we said before, in cythroma. Okay, occurred by reaction between NADBH2, not all NADBH2, also only one H2 hydrogen, okay, that carried on NADB, okay, and used in fixation of also carbon dioxide. So NADB fix hydrogen, hydrogen fix carbon dioxide gas and reduce it to turn it to carbohydrates. Okay, Melvin Calvin and his associates in California University revealed together the nature of dark reaction. Okay, so Melvin Calvin, okay, scientists need to know the nature of the dark reaction and use isotope also, but not here oxygen but carbon radioactive 14. Okay, this is a Calvin cycle. Okay, carbon dioxide here get to cythroma, okay, and reduced by hydrogen gas to give water and glucose. Okay, Melvin Calvin, okay, made his experiment on chlorella algae. Okay, this apparatus shown the dots chlorella algae, okay, and bomb for carbon dioxide isotope carbon with isotope carbon, and we have here hot alcohol, and using light for making photosynthesis process. Chlorella algae is green algae, okay, so it contains chlorophyll to make photosynthesis process. Okay, first one, they placed the chlorella algae in apparatus and supplied it with carbon dioxide, as we said. Okay, carbon dioxide with isotope or radioactive carbon 14. Lamp was light, and the chlorella algae then immersed in beaker containing hot alcohol. Why we immersed chlorella algae after a while from exposing to light to inside a beaker containing hot alcohol? Because, or two, stopping all functions and all biological process inside the chlorella algae, and also stop photosynthesis process. Number two, by special means, okay, Melvin Calvin and his colleagues used some special means to separate the first product from photosynthesis process, the first stable one, okay, and by observation he detected the type of carbohydrate that first produced was compound named by Big Al or phosphoglyceraldehyde. Thank you and goodbye.